1. We are going to see genetic variation in bacteria. It can be take place by using mutation and gene transfer. There is the results of gene defect, alteration in transcription, amino acid sequence functions, example antibiotic resistance, lethal undetected mutation. There are different types of mutation: base substitution, that is replacement mutation; deletion, insertion, inversion, duplication, which is common type of mutation. Gene exchange are of three types, that is transformation, transduction, and conjugation. Transformation is the fragment of exogenous naked bacterial DNA are taken up and absorbed into recipient cell. It's common in influenza, and Streptococcus pneumonia. Here is the diagram which representing the transformation. There is a bacterial cell containing bacterial chromosome. There is exogenous fragment of bacterial DNA is taken inside the cell. Then it combined with the bacterial chromosomal DNA and multiply or passes to the next generation. Another one is transduction, which is phase-mediated transfer of genetic information from donor to the recipient cell. For example, beta-lactamase production in S. aureus and toxin production in Corynebacterium diphtheria. This cell, where you can see that there is a phase particle containing DNA fragment from the host bacterial chromosome, that is phage DNA. Uh, there is a viral capsid. Inside this there is a phage DNA. These are the phage particle. They are allowed to infect the bacterial cell by means of lytic or lysogeny cycle. The phage DNA is now enter into the bacterial cell and combined with the chromosomal DNA and transferred to the next generation. The third term is conjugation. The major way bacteria acquire additional genes, that it's plasmid mediated. Cell contact required and gene residue on plasmid resident within donor cells transferred to the recipient, that is female cell. Conjugation can be done by mating. Conjugation among gram-negative mediated by plasmid called F factor, that is fertility factor. Gene encode changes in surface by producing a sex pillar. This facilitates the capture of F minus cells and the formation of a conjugation bridge through which DNA passes from F plus cell into the F minus cell. In this cell you can see that there is a sex pillar present on the bacterial cell, and plasmid is also present in the cell. There is one F plus cell and another one is F minus cell. There is villi formation in between both the cell. By means of this bridge, the F plus cell transfer plasmid into the F minus cell. Now there is only 50% plasmid is transferred to the next cell. So they have their own plasmids. So both the cells having the same plasmid. Conjugation among the gram positive, it involves clumping of the cells and pheromone secretions. Genetic recombination. After gene transfer, there are three possible results. First is exogenous DNA degraded by nucleus. Second is stabilized by circulation become plasmid. Circulation become plasmids. Form a partially hybrid chromosome with segment derived from each source. Thank you.